Okay. All right, so hi everyone, welcome to week 3 for BCH 210. Now today is a little bit special because I'm going over the lecture content, but I'm also going to go over some examples for your PDB assignment where you have to make connections to the course material, which is something Patterson really likes. So if you write a lot of connections, she's going to be happy and she'll mark your assignment easier and you'll get a higher mark. Okay, so I'll go through those at the end of the session after I've covered everything for the lecture materials. All right, so just a note for this week, you only have 2 lectures, right? So it's on studying protein structure and then studying protein function. Then the third, sort of semi-lecture from your TA, that one is not going to be testable material. So you only need to know the first 2 lectures for your quiz and your BBQs. But I actually highly recommend you go watch her lecture, so your TA's lecture, because the way she explains things is in a way where she makes connections to the lecture content. So just listen to what she says and you might be able to get some hints as to what you need to write or what kind of connections you need to make for your PDB assignment, okay? Right, so with that said, let's get started. So last week, right, we were talking about this section here. So how do you isolate your protein of interest? So now you've isolated your protein of interest and then you want to study it. So how do we study this protein? And we're going to start off with talking about how to study the structure of our isolated protein, okay? Now, structure, as you know, is important for the function of a protein. So if we understand structure, we can help to understand the function of a protein and how it is able to do its job, okay? Now, obviously, we have 4 levels of protein structure, and sequencing is going to give us our primary protein structure, all right? So then how do we determine the secondary structures and the 3-dimensional tertiary and quaternary structures? And those are the techniques we're going to cover today, okay? So the first method is tryptophan fluorescence spectroscopy, okay? So in order to understand how this particular technique works, I actually want to first cover the basics on fluorescence, just so that everyone is on the same page, okay? So with fluorescence, what we have are these things called chromophores, and sometimes they're also called fluorophores, okay? And these are compounds that essentially have these conjugated double bonds, which creates resonance in the structure, which means their electrons are delocalized, okay? So these delocalized electrons is able to absorb energy in the form of light, and specifically, it will absorb energy in the form of light of a specific wavelength, okay? So here I have included a spectrum of light with the wavelength on top. So red light has the longest wavelength, 700 nanometers. Purple light has the shortest wavelength, 400 nanometers. Anything shorter than purple is going to be UV, and anything longer than red, visible light, is going to be infrared, okay? So your delocalized electrons, depending on which fluorophore or chromophore you're talking about, they're going to absorb energy from light of a particular wavelength, okay? And we call this wavelength the excitation wavelength, which is written right over here, okay? So this purple excitation wavelength comes in, the electron absorbs this energy, it's going to jump up to the excited state, which is very high in energy, but this state is very unstable. So the electron is then going to jump back down to the low energy ground level, all right? And as it's jumping down to this ground state, what's going to happen is it's going to release energy in the form of light of a longer wavelength, right? So the wavelength symbol is indicated with this, right? So this is wavelength. Okay, so the wavelength of the light that is emitted by a fluorophore is always going to be longer than the wavelength that is used to excite that fluorophore. Okay, so keep this in mind. So coming back to this technique of tryptophan fluorescence spectroscopy. So we've seen tryptophan before. It's a quite special amino acid when it comes to different techniques that scientists use. We previously discussed how it is able to absorb light of 280 nanometers, and then we can calculate the concentration of protein based on this absorbance value. But the indole ring in the side chain of tryptophan, as you can see, has that benzene ring, right? So these are, if I draw the double bonds, it's like this. This whole ring has conjugated double bonds. So they have delocalized electrons in the indole ring that can be excited by light and emit light in response. Okay, now it is specifically going to be excited by light in the UV range. So we're talking about light with wavelength of 270 to 295 nanometers. And then once it's excited, it's going to emit light of a longer wavelength, all right? And the specific wavelength of the light that is emitted is going to depend on the environment around that particular tryptophan molecule, okay? So if this tryptophan is, say, for example, in a hydrophobic environment, right? So somewhere, say, buried inside of the protein, right over here, this blue guy here, okay? The emitted light, the wavelength is going to shift towards a shorter wavelength, towards a 310 nanometer range. And we're going to call this phenomenon a blue shift. So the light is blue shifted, right? So there is a decrease in the wavelength of the emitted light if tryptophan is in a hydrophobic environment. And additionally, the intensity of the light that this tryptophan is going to emit is going to increase. So indicated by this high peak in this graph, okay? Now, if tryptophan is instead in a polar environment, okay? So for example, at the surface of the protein indicated by this little red guy here. Now, at this particular polar environment, the wavelength of the light that is emitted become red shifted. So it shifts towards a longer wavelength, towards 355 nanometers. And the intensity of that emitted light is also going to decrease, okay? So you need to know at which point is tryptophan going to blue shift versus red shift. So under which environmental conditions will tryptophan blue shift versus red shift, okay? Now, obviously, this can be used to monitor the environment that's around tryptophan, right? But it can also be used to monitor conformational changes. So I've written it here for you. So we're talking about something like protein folding, for example, as conformational change. So you can imagine that as a protein is unfolding, so it's falling apart, all right, the tryptophan that are initially buried on the inside of this protein are going to get exposed to the polar environment, to water, right? So this particular tryptophan is going to undergo red shift as an indicator of protein unfolding. Now, if you take this unfolded protein and then you allow it to fold back into its native structure, this tryptophan that was exposed to the polar environment gets refolded back to be buried on the inside of this protein, and its emission is going to undergo a blue shift towards 310 nanometers. Okay, so one of the reagents that I want to introduce is urea, right? And urea is going to cause your protein to denature. So it's going to cause your protein to unfold. So occasionally you might see a question that uses urea as an example. Just know that this is a denaturing agent. So if you add urea, your protein is going to unfold. okay? So that's tryptophan fluorescence spectroscopy. It's used for determining the local environment around an amino acid, also for monitoring conformational change in the form of protein folding and unfolding. So then we have a second method. This is called IR spectroscopy, IR for infrared light, okay? So this particular technique relies on the ability for peptide bonds to absorb light in the IR wavelength, so the infrared region of light, okay? The reason why peptide bond is able to absorb IR radiation is because this bond is constantly in motion. So unlike what I've drawn here, they're not stable. So for example, this carbonyl bond can stretch, okay? And this amine bond over here can undergo bending. So a little bit of bending. So these tiny little movements is what allow the peptide bond to absorb light, specifically infrared light, okay? Right. So these movements are being measured in our spectroscopy, and the ability for them to absorb infrared radiation depends on hydrogen bonding that are present in the secondary structure of proteins. Right? Remember, hydrogen bonding between backbones is what creates secondary structure, all right? So by having a hydrogen bond indicated in red here, this hydrogen bond is actually going to restrict the motion of the peptide bond. So it's going to restrict the stretching of the carbonyl and the bending of the amine group, okay? And this is detected in the spectrum, right? Now, one thing I will point out is if you have a strong hydrogen bond, it's going to mean a weaker covalent bond, right? And that's just because this hydrogen bond is essentially pulling things towards it, and it's weakening the strength of these covalent bonds, okay? So obviously different kinds of hydrogen bonds between different amino acids are being formed in different secondary structures. So if you look at the IR spectroscopy that corresponds to each of these secondary structures, you'll see there is quite a dramatic difference. Okay, so here is a spectrum. We have the beta sheets in blue, alpha helix in red, and the random coils, which are just secondary structures that do not belong to either category, they're in green, okay? Now, obviously with this technique, you can run your protein through IR spectroscopy, and it's going to tell you the percentage of each of these secondary structures that you have, okay? It's going to tell you the percentage of your secondary structures, as well as let you know about the hydrogen bonds that are present, because hydrogen bonds will affect the peptide bond's ability to absorb infrared radiation. Now, I will point out that even though you get to know the percentage of secondary structures that are present in your protein, this method is not going to tell you where those secondary structures are located. So what you need to know is the primary amino acid sequence, and you're looking for stretches of amino acids that prefer one of those secondary structures. So if you're looking for a beta sheet, you're looking for amino acids that are either aromatic or are beta branched. And then you can sort of correspond that to your findings from our spectroscopy to get an idea of where those secondary structures are actually being formed in your protein, okay? And then we also have circular dichroism, short-term CD. And this is also used to determine the secondary structure of proteins, so the percentage of alpha helices versus beta sheets versus random coils. And similarly to IR spec, it's going to tell you the percentages, so the composition, but they're not going to tell you where those secondary structures are, okay? Now, with circular dichroism, what it relies on is this alpha carbon right here. And specifically, it relies on this alpha carbon being chiral. So out of the 20 amino acids you learned, 19 of them have an R group that is different from the rest of the groups attached and therefore have a chiral center for its alpha carbon. The exception is obviously glycine, right? Glycine has that hydrogen for its side chain, and it doesn't have a chiral center for its alpha carbon, okay? So for the amino acids with the chiral center, right, this chiral center is able to rotate light, and specifically it's able to rotate circularly polarized light. So this incoming light over here is essentially, rotating, okay? And then once it hits this chiral center for the amino acid, the rotation is going to change. So for example, in the one I've drawn, right, this red rotation got turned 360 degrees, and now it's rotating in the opposite direction, okay? So this is what circular dichroism detects, is this change in the rotation of that circular light, okay? And because of this, the secondary structures all have different bondings, different chiral centers, they have specific amino acids that prefer to be in them versus some other secondary structure. They are going to be able to rotate this light in different ways, okay? And this is summarized in this little spectrum chart over here. So with the beta sheet, okay, shown in blue, it's going to have a minimum at 270 nanometers. So we indicate the minimum with the minus sign. For the alpha helix, it has a maximum, so positive at 195 nanometers. And it has two minimums at 208 and 222 nanometers respectively. With a random coil, you have one minimum at 198 nanometers, okay? Now, with circular dichroism, we said it can be used to determine the composition of your secondary structures, but it can also be used to monitor any conformational changes. So once again, we are talking about the protein unfolding and then folding back, okay? So as conformational changes are happening, right, you have, say, protein unfolding, you have your beta sheets and alpha helices starting to unfold, and they're going to look more like random coils as the protein unfolds, right? So you will see a dramatic change in this spectrum for your overall protein, where the spectrum is going to look more and more like the random coil spectrum than what it looked like before, okay? Now, obviously, if you reverse this process and you fold your protein back together, the random coils are going to start resembling alpha helices and beta sheets more, and you'll be able to see this reflected on a change in the CD spectrum, okay? So we've talked about secondary structures, but what about the tertiary and the quaternary structure? So the final three-dimensional structure of your protein. Well, we have three different techniques that can be used. We have X-ray crystallography, NMR, and cryo-EM, okay? The most popular method right now by far is X-ray crystallography. However, this method is very particular because it requires your protein to be able to form these really high quality, very ordered protein crystals, right? And typically how we form these crystals is similar to how you elute proteins off of the ion exchange column, is by making them neutral, okay? So this can be done by increasing the salt concentration. So these salt ions, for example, Na plus and Cl minus, can come to your protein, right? Bind and neutralize any of the charges on your protein to make your protein overall neutral. Or you can change the pH. Specifically, you can make the pH within 1 unit of the isoelectric point of your protein, right? And within that 1 unit, your protein is going to be neutral, overall neutral, okay? Now, you don't want this to be exactly equals to your PI, because at that point, your protein is going to be all neutral and they're going to all start aggregating together, and they won't be able to form a good quality crystal, okay? So you want your protein to be neutral to be able to come together slowly to form this crystal, but you don't want them to be so neutral that they form aggregates or these little random blobs instead of a crystal, okay? So you take a crystal that you formed using one of these methods, and many of you would have seen this in your PDB assignment, okay? You take this little crystal here, and then you shine an X-ray onto that crystal. And then the electron clouds that are in this crystal is going to cause this incoming X-ray to become diffracted into a specific pattern. And then the computer will detect this diffraction pattern, translate it into an electron density map, and then you can start fitting atoms into that electron density map. And this will tell you the exact location of each individual atom, okay? Then you combine this with your knowledge of protein sequence to obtain the final three-dimensional structure. And with X-ray crystallography, the resolution of your final structure is very, very high. So it's 2 angstroms or smaller. So this is a golden standard for biochemistry, because at this resolution, you can see the smallest atoms there, and they are the hydrogens, okay? So X-ray crystallography, finicky because it requires protein crystals, but it will give you a very high resolution image. Now, I will point something out that might relate to some of your PDB assignments, okay? So X-ray crystallography requires these really good protein crystals, but sometimes, even though you formed a really good protein crystal, there can be areas inside that crystal that's a little bit flexible, okay? So they're moving around a little bit. They're not very ordered. You can have little areas in that crystal that are not ordered, okay? And when they're not ordered, it's going to cause a problem for the X-ray crystallography method. And these areas are going to become lost, right? So it's not going to form a defined diffraction pattern because they're moving around constantly. So the computer can't decipher that pattern because it looks like nothing to the computer. And it won't be able to generate an electron density map for that region. So you actually end up losing the amino acid in that particular area that's really flexible. So imagine if you have an N-terminus on your protein that has a lot of flexible amino acid, it might not be able to crystallize very well in terms of that particular N-terminus region, which is why with crystallography, you might actually end up losing the first amino acid or even the first, say, 10 amino acids from that N-terminus because it's too flexible to be detected by X-ray crystallography, right? So something to keep in mind for your PDB assignment. This could be one of the reasons why the first amino acid is not found at your N-terminus, okay? Now, right? Okay. Yes, question.
Yeah, I have a little bit question about the flexible area. Because this area is flexible, we cannot see anything on the the form. Is there any possible some ions bind to this area because we cannot see exactly the structure, but the ion just like inserted inside?
I mean, well, I would say it depends, right? So if the ion is bound to this flexible area, it depends on how flexible the ions are going to be. Okay, so if the ions are also flexible and it's moving together with this flexible area, it's also not going to be detected via X-ray crystallography, and it also gets lost, right? However, if the ion itself, even though it's bound to this flexible region, if the ion can form an ordered crystal, which ions typically are able to do, right? You'll detect the ion, but you won't detect the amino acid that the ion has bound to. So you'll still see the ion as part of your structure, you just don't know what it's interacting with, okay? But it might actually give you an idea. So if you see a positively charged ion sitting in this location with no amino acid, it's probably interacting with the negatively charged amino acid. So that's the ion issue. Yeah.
Cool, thank you.
Yeah, same thing with ligands, right? If you have a really big ligand, sometimes it's not able to form a structure, form a crystal, which is why, the authors might end up using some other technique, or the authors might choose an analog of that ligand. So something that looks like that ligand, but is actually able to form crystals with the protein, okay? So another thing to consider when you're writing the PDB assignment. All right. So what if your protein can't crystallize, right? What if you form a crystal, but the crystal is not very good quality and you can't use X-ray crystallography to determine the 3D structure? What do? Well, you can rely on NMR and cryo-EM, okay? So let's talk about NMR first. This stands for nuclear magnetic resonance, and this is an imaging technique. And this is really, really good for very small proteins, so less than 25 kilodaltons in size, and they are for proteins in solution, all right? So these are soluble aqueous proteins floating around in the solution, and it's still flexible, right? It's still able to undergo conformational change, unlike the ordered proteins in a crystal, okay? So what NMR does is it takes a solution of proteins and it will expose these proteins to electromagnetic radiation. So this little symbol, this stands for electromagnetic radiation, okay? And the protein is able to absorb some of this electromagnetic radiation, and it's going to transform this energy into motion in the form of nuclear spin. And specifically for NMR, we are looking at the nuclear spin of hydrogen 1, nitrogen 15, and carbon 13. Okay. Now, different functional groups in different environments will produce a different NMR reading. Okay. And this is what is used to then construct a 3D structure, because you can sort of tell what the functional group is that's generating this nuclear spin and what environment is in, okay? All right, so with NMR, sometimes you do have to rely on 2D and 3D NMR because this technique is lower resolution than X-ray crystallography. So it is greater than 2 angstroms, which means it cannot see any of the hydrogen atoms. But you can actually help increase the resolution by combining the nuclear spin of different nuclei, okay? So 2D NMR looks at hydrogen 1 and nitrogen 15, and then 3D NMR also includes carbon 13 in it, okay? Right, so because this is done using proteins in solution, these proteins are soluble, they're still flexible, they're no longer fixed in place like in a crystal. You can use NMR to monitor conformational changes in the protein. So for example, protein folding and unfolding, because the protein is still flexible in solution, okay? Now, what happens is you will see a change in the NMR spectrum as a protein folds or as it unfolds, okay? And you can actually also look at binding interactions between your protein and, say, a glucose molecule or any type of ligand, because you are able to detect this conformational change that's happening, so you can see how your protein changes shape as it binds to that specific ligand. Okay, So this is something that NMR can do, but X-ray crystallography cannot, okay? All right, so the other technique was cryo-EM, which stands for cryo-electron microscopy, and cryo just means cold, and you'll see why, okay? So this particular method is really good for very large proteins or large protein complexes, so quaternary structure proteins, greater than 100 kilodaltons in size. And once again, it is for proteins in solution, so you do not require a protein crystal. However, in this case, your protein in solution is going to be frozen very, very quickly using ethane, specifically liquid ethane. So by freezing them within a few milliseconds, it helps to preserve the structure of your protein without having the protein to crystallize into a crystal, okay? So it's still protein in solution, but you freeze it very, very quickly, and this is going to preserve the structure of your protein so you can study the structure, right? Now, because it is frozen, right, even though your protein is in solution, it is no longer flexible, okay, which is why cryo-EM cannot be used to detect conformational changes, unlike NMR, where the protein is still able to change shape. Right, so with cryo-EM, you take this frozen sample, right? And the good thing here is you only need a very little amount of that liquid that you freeze. Okay, you take that frozen sample, you shine a beam of electrons onto that sample. And then the nucleus of different atoms are going to diffract that electron beam into a specific pattern that is then detected by the computer and used to generate the three-dimensional structure. So the theory is quite similar to X-ray crystallography, right? You have an incoming beam of light, and then you're having that light being diffracted by your protein, okay? But the difference is, with cryo-EM, it is the nucleus that's diffracting the electron beam, versus if we come back to X-ray crystallography, it's the electrons that are diffracting the beam. So I know that Dr. Patterson mentioned that in theory, cryo-EM can give you higher resolution than X-ray crystallography. However, in reality, because this technology is relatively new, is much newer than X-ray crystallography, the resolution, while it's high, is not that much higher than X-ray crystallography at this point in time. But then this technology is constantly getting better and better, and eventually we're going to be able to surpass X-ray crystallography with cryo-EM in terms of resolution, right? So in summary then, right, this is our diagram for all of the techniques that can be used to study protein structure, okay? So in summary, you have infrared spec, you have circular dichroism. Both of these can be used to study the secondary structure of a protein, right? Tryptophan fluorescence plus circular dichroism plus nuclear magnetic resonance. Those are the three techniques that can monitor conformational changes in your protein, so changes to the structure of the protein. And then you have the X-ray, the NMR, and the cryo-EM that are used to elucidate the three-dimensional structure. So make sure you know that what each of these methods are used for. You're going to get questions where she'll ask, oh, I want to determine, say, the secondary structure of this protein. Which of the following techniques should I use? You should immediately think infrared or circular dichroism, okay? And I have a table that summarizes all of these techniques in terms of, what they're used for and the theory behind them in a little table in the notes and questions PDF file. So if you're someone who prefers a table version, it's there for you to review, but the content is the same. So I'm going to now switch to the questions that are related to studying structure, right? So coming to this one, and let me just check. Can you guys see the Word document? Okay, cool. Right, so we're just going to walk through some of the very simple questions that's related to this section. Right, so the first question is, what property of tryptophan allows it to fluoresce? And because I want to cover the PDB assignment at the end, I'm actually just going to walk you through the questions. So you can try them again on your own, but I will just walk through them really, really quickly. Okay, so tryptophan, fluoresce. Fluoresce meaning to emit light, right? So we've mentioned in the little summary that the reason why tryptophan is able to fluoresce is because of its indole ring, so its side chain, and specifically because its side chain has conjugated double bonds, which means that electrons are delocalized in its side chain. So the answer to this question is C, okay, and you can double check this yourself as well. Now, the second question is actually a really poorly written question. Okay, and this was taken from a previous exam, but let's just go through this. Okay, so you've isolated a novel metabolic protein. I don't care what kind of protein it is, it's a new protein. You want to understand how its structure plays a role in its function. What do you want to determine? The secondary structure and how its interaction with glucose molecules can affect its structure. And then the question asks, what sequence of techniques would you use? So we're actually asking you, which of these techniques would you use, and in what order would you use? Now, here is the reason why this question is written so badly. Because it's not explicitly stated which one you want to find first, right? Do you want to find the secondary structure first, or do you want to look at interaction first? So with this question, if you see something like this, you need to assume that whatever is written first is what you want to look at first. So you want to first look at secondary structure and then look at interaction with glucose, right? So if we're trying to look at secondary structure, I mentioned the two techniques that are used, IR spec and CD, right? So we can right off the bat eliminate C and D because the first step doesn't involve a technique that can be used to determine secondary structure. And then we look at the second part, interaction with glucose. So we're talking about binding interactions with a ligand. And you should immediately think NMR is the only technique that can be used to do this. So the answer to this question would be B. Now, this question is probably not going to show up on a quiz just because I think they realize that it's really confusing. But you might get a similar question where she just asks you, oh, you want to determine the secondary structure. So she take out this half of the question and then give you, say, instead of IR, she'll give you X-ray crystallography here. Then in this case, obviously the answer will be circular dichroism, okay? So a simpler version is probably what you will see on either your BBQ or the quiz, okay? Now, the third question is this. You want to study the stability of luciferase, this is an enzyme, which contains a single buried tryptophan residue. So you think buried tryptophan, that's hydrophobic, right? And hydrophobic tryptophan has blue shifted emission wavelength. So increase intensity, decrease wavelength, okay? So what are you doing then? You are adding urea. Remember how urea, I've introduced it as a denaturing agent that's going to cause your protein to unfold, okay? You add urea to your protein sample, and then perform tryptophan fluorescence spectroscopy as well as circular dichroism, what would you see? Okay, so we're looking at the unfolding of a protein. Okay, we've said that circular dichroism can be used to monitor conformational changes in your protein. And this conformational change is going to be indicated by a change in the spectrum for CD. Okay, so you look at the second half of these answers. CD spectrum, we know, will change as the protein changes shape. So we're going to take off A and take off B as incorrect. Right? Now we look at tryptophan. So tryptophan was initially buried inside the protein, but as the protein unfolds, it's going to become exposed to the polar environment, that being water. So tryptophan is now in a polar environment, and in a polar environment, tryptophan is going to undergo a red shift, and red shift involves an increase in wavelength and a decrease in intensity. So we're going to look for that. Fluorescence will decrease, so that's a decrease in intensity, and redshift. So your answer would be B for this question. Okay, so now I'm going to go back to our diagrams, and I'm going to talk about the techniques regarding protein function. Okay, and here is where things gets a little bit more complicated. All right. Okay, so you studied the structure of the protein. What if now you want to study the function of this protein? So we know that protein function involves forming mostly non-covalent interactions, right, with other proteins or other molecules. Okay, and then the structure of the protein is what mediates its ability to form these non-covalent interactions, because it's the functional groups on the side chain of amino acids that are going to form non-covalent interactions with functional groups on other proteins or on other molecules. Yeah, no worry about needing to leave early. Feel free to leave early, it's recorded anyways, okay? But do make sure that you watch the recording, because there will be a portion about the PDV assignment as well, okay? Right, so back to this relationship. Structure is important for function because structure mediates what non-covalent interactions can be formed with other proteins and other molecules, right? And since structure is so important for the function of the protein, when you're studying the function of the protein, you need to make sure that your protein that you're using is folded into its correct final shape, right? If it's incorrectly folded or if it's unfolded, it's not going to do its proper job, right? And then you'll get a weird result, okay? So in order to maintain the structure of your protein, what you can do is isolate your protein and then put your protein into a buffer that will mimic the cellular environment that the protein is normally found in, okay? So if it's normally found in the cytosol, you will choose a buffer with a pH that mimics the cytosol, okay? So this will be done if you're doing an experiment that is considered an in vitro experiment, which means you're running it outside of cells. So you're breaking the cells open, you're taking the protein out, and then you're performing some sort of experiment on those proteins. However, maybe you don't want to do that. Maybe you don't want to break open the cell and risk disrupting the structure of the protein. So what you can also do is run an experiment in vivo, so inside actual cells. So in this scenario, right, the protein is already inside the cell. So you are mimicking a cellular environment in a sense, because it's not outside of the cell. So you don't have to worry about, pH conditions or buffer composition, okay? All right. So there are actually many different kinds of methods for studying protein function, depending on what function you're looking at. But for BCH210, because we introduced this concept of the relationship between structure, function, and interactions, we're going to talk about the methods that are used to identify whether or not proteins are interacting or whether or not protein is interacting with other non-protein molecules. Okay. Now, this is not easy. Okay, studying these interactions is very difficult. And the reason for this is because most of these interactions are non-covalent. And we know non-covalent interactions are very weak interactions and they're reversible interactions, all right? So these are going to be very transient and short-lived. They're going to form and then they're going to break apart and then they're going to form again and then break apart. So this makes it really difficult to actually study these interactions, okay? Now, additionally, to add another layer of difficulty, right, most proteins and even molecules are colorless compounds in nature. So you can't actually visually see the interaction happening. So you need now another method, and this time a visualization method, in order to actually see those non-covalent interactions being formed, sort of, okay? Just an indicator that this interaction is happening. And with visualization methods, we typically use something called a reporter. So this could be a protein, this could be some non-protein molecule, but generally reporters are going to be protein molecules that will provide you with a visual signal only if the interaction is occurring between your protein and another protein or another molecule, depending on what interaction you're looking at. So if they interact, the reporter sends out a visual signal that you can then detect, and this will tell you, hey, these things are interacting, okay? So reporters can be things like fluorophores that emit light, as we've learned. It can also be things like enzymes. And we'll see an example of a reporter being a luciferase enzyme, and I'll talk about what it does when we get to it, okay? All right. Now, yeah, that is all I wanted to mention for that part, right? So let's get down to the methods that can be used. So one of the methods that can be used to study protein-protein interactions is this thing called a pull-down assay, okay? So what pull-down assays do is you're essentially purifying protein complexes, okay? So you're purifying proteins alongside with whatever that protein is interacting with, because its interactors are going to be bound non-covalently to your protein of interest. So theoretically, if you use a condition that maintains those non-covalent interactions, when you isolate out your protein of interest, everything that's bound to your protein is also going to come down with it. And then you can then identify whatever comes down together with your protein to identify the interactors, right? So with this particular method, we do need to use a very mild condition as to not covalent interactions. This can be difficult sometimes depending on which cells you're using and because you need to break open the cells, okay? So in the process of breaking open the cells, it can be very easy to actually break apart those non-covalent interactions. And then your pull-down assay turns into a simple chromatography experiment where the only protein you're going to get out is your protein of interest because you've disrupted all of those non-covalent interactions that allow the interactors of that protein to stick to that protein, okay? Now, here's a specific example of a pull-down assay, and it is called co-immunoprecipitation, also known as co-IP for short. And the reason why this is called this is because we use antibodies. So this immuno part indicates the use of antibodies, okay? So in this particular experiment, we're trying to identify whether or not two proteins interact. So two specific proteins, do they form non-covalent bonds? So let's call these proteins A and B. So do A and B interact? So this is a question you'll be trying to answer with co-IP. So what you have to do is you have to get an antibody that is specific to one of your proteins of interest. So it's going to bind to one of these two proteins. So let's say it's going to bind to protein A. Right? So you attach your antibody onto a solid support, for example, a bead column similar to affinity chromatography, right? You attach it to this solid support. You take your cells, break them open, remembering to use a condition that will not disrupt non-covalent interactions. Right? You take that crude extract, pour it down this solid support column, and hopefully your protein A will get bound to the column. And if your protein B is interacting with protein A, it's also going to get captured because it's together with protein A. Right? So then you break this, oh, sorry, not here, but you break this interaction between the antibody and protein A, elute this out, and then you can perform Western blotting using the antibody versus the other protein. So in this scenario, the antibody is going to bind to protein B, okay? And then this will tell you whether or not A and B interacts, because if they do, B gets pulled down together with A, and you'll detect protein B on your Western blot, okay? Now, this does require you to have antibodies specific to your proteins of interest. So that is also something to keep in mind. All right. Now, I mentioned this issue with non-covalent interactions being really transient and being really easily disturbed and disrupted. Okay, so sometimes it can be really hard to maintain these interactions, okay, especially when you need to break cells open. So one of the ways in which you can actually stabilize these non-covalent covalent interactions is through the use of chemical crosslinkers. So this will allow you to capture those protein complexes that are forming those non-covalent interactions without having to worry about breaking those interactions apart. Okay? The reason why crosslinkers allow you to do this is because these crosslinkers will actually form covalent bonds, right? So very strong, essentially irreversible bonds between the functional groups of amino acids to capture the complexes together. So it's going to stabilize the structure of that complex. And the stabilization function of chemical crosslinkers is actually oftentimes used even for X-ray crystallography in order for the protein to become stable and therefore be able to form those very ordered high quality crystals that are needed. Okay. So here are just some of the examples of amino acid functional groups and what they can form interactions with, okay, and form covalent bonds in this case with. So primary amines, so this would be your nitrogens, all right, can form covalent bonds with aldehydes and carboxylic acids as well, okay. Hydroxides was something that Dr. Patterson mentioned, and this is just two nitrogens linked together, okay? Hydroxides are not as fussy as amines. Hydroxides can form covalent bonds with aldehydes, carboxylic acids, as well as any carbonyls, so ketones, okay? And then we have our thiol groups. So our SHs on methionine and cysteine. And with disulfide bridges, we are specifically talking about the cysteines because they have the SHs at the very end of their side chain, okay? So cysteines can form disulfide bridges using these thiol groups, but in order for this bridge to form, you need oxygen under normal conditions, okay? However, you can actually use chemical cross-linkers to force cysteines to make these disulfide bridges. So here are the two examples that were introduced, and you should probably know them. Okay, so maleimides and iodoacetamides. These are chemical cross-linkers that will forcibly make these disulfide bridges between cysteine molecules in order to link the cysteine molecules together. Okay. Now, with this specific example of maleimides and iodoacetamides, they specifically target the thiol group. So they specifically force the formation of a covalent bond between the thiol groups known as a disulfide bridge, right? But chemical cross-linkers can also be non-specific. So meaning it doesn't really care about which functional group is going to form a covalent bond for. It's just going to form covalent bonds between many different functional groups, okay? So an example of a non-specific chemical cross-linker is formaldehyde. And because it's non-specific, it doesn't depend on the composition of amino acids. So it doesn't depend on, say, the cysteine residues if you're using an iodoacetamide, which is the reason why formaldehyde in a lab setting, you'll see we use this guy a lot as a fixative agent. So we add it to our cells, and it will fix everything in place. So if protein A is interacting with protein B, it's going to make those covalent bonds between those proteins and then keep them together, okay? Now, there are also photoactivatable cross-linkers. So there are many, many different kinds of cross-linkers. And let me write this out for you. So photoactivatable cross-linkers. As indicated by the name, it is activated by photons, so it is activated by light. So only until you expose this chemical cross-linker to light, for example, visible light or UV light or infrared radiation, then it is able to form these covalent bonds. If there is no light, it cannot form covalent bonds, okay? And in addition to having photoactivatable cross-linkers, non-specific cross-linkers, specific cross-linkers, we also have heterobifunctional cross-linkers, right? So these are chemical cross-linkers that have many different functional groups attached to it, and each of these groups have a specific function. So the cross-linker by itself has multiple different functions, hence heterobifunctional. In this particular word, 2 functions, but it can also have more than 2 functions, okay? All right, so for example, with a heterobifunctional chemical cross-linker, you can have different reactive groups that will form covalent bonds between different amino acid functional groups. Okay, so you can have one, for example, that is iodoacetamide that forms the disulfide bridges between cysteines. You can have a different chemical group attached to the same cross-linker that will, say, for example, react with amides to form a covalent bond, right? You can have a purification tag attached to your chemical cross-linker, right? So something like biotin for easy purification of those protein complexes. You can have a reporter molecule that's attached to your chemical cross-linker for you to visualize where those interactions are, or to visualize those protein complexes. And finally, you can also have a spacer of defined length in your chemical cross-linker. And what the spacer will do is allow covalent bonds to be formed between amino acids within the distance that is the length of the spacer, right? So it's used to determine the proximity of functional groups. So how close together certain functional groups are, right? So those are pull-down assays, chemical cross-linkers. Now, what if you want to instead study the interaction between different proteins inside of the cell? Okay, so you don't have to worry about needing to mimic the cellular environment in order to maintain structure. Well, what you can do is use fusion proteins, right? So obviously, if you're looking at protein-protein interactions inside the cell, you come across a problem where you can't actually visualize the proteins by themselves because they're colorless. So you actually need to attach a visualization tag or something that can tell you the interaction is happening to these proteins. And we do so by fusion protein technology. So this is something that I sort of introduced for the PDB assignment because it relates to cloning, right? So to make that fusion protein, what you need to do is get the gene for that protein, and then you're going to modify that gene by attaching a DNA sequence that encodes for a specific tag that you want to add. So in our PDB assignment, I said it could be a histidine tag for purification, right? So this DNA encoding for that tag can be for detection, but it can also be for purification like the histidines. So this modified DNA with the two genes, essentially, is what we call a recombinant DNA molecule. Okay? So we take this recombinant DNA, we stick it into a plasmid, and then we stick this plasmid into a cell. And then the cell is going to start transcribing this modified recombinant DNA into mRNA and then translating that into your fusion protein. Now has your protein of interest attached to this tag that you've included, okay? So this is how we make fusion proteins. All right, so coming back to these methods that study protein-protein interactions in cells using these fusion proteins, we're going to kick off with the two hybrid system. Okay, so here, what you want to look at is whether or not two specific proteins will interact. So once again, let's call these proteins A and B. So do protein A and protein B interact? So in a two hybrid system, you're going to need to make two fusion proteins, one for your protein A and then one for your protein B. So what happens with this method is you're going to add a bait tag to one of the proteins and then add a prey tag to the other protein using fusion protein technology. Okay, so what these baits and preys are essentially proteins, or they can be parts of a protein. So for example, a domain in a protein or a subunit of a multimeric protein. And bait and prey, if they are very close together, they will do something that can be detected as an indicator of interaction, right? So bait and prey will do something special only if they're very close together, okay? So what you're looking for is, what you're looking for as an indicator of the interaction between A and B, right, is that special something that the bait and prey are going to do once they are in close proximity. Okay, so here is an example, a very common example for a two hybrid system. Okay, so in this scenario, your bait and prey together are going to form a functional transcription factor, right? So these transcription factors are what's responsible for transcribing DNA. So if you have an interaction occurring between A and B, right? A and B interacts, they come together. Because the bait and prey are attached to A and B respectively, they also bring the bait and the prey into close proximity, okay? So something is going to happen. In the case of transcription factors, your bait and prey together are going to go find a reporter gene. It's going to start transcribing that reporter gene into mRNA, which is then translated into protein. And you're going to detect this reporter protein, and this is going to indicate to you that yes, A and B are forming an interaction, all right? So this reporter protein could be, for example, a fluorescent protein, say a green fluorescent protein. And then if you see the green fluorescence, that means A and B has interacted. They brought the bait and prey close together in order for this reporter protein to be able to be made by the cell, okay? Now you can also have something like this. So this is a system where not only do you have the bait and prey attached to your respective proteins of interest, you also add the reporter protein directly to one of these proteins that you want to study. And pardon me, this red one should actually be protein B, okay? So in this scenario, A and B, say, interacts, they come together, right? This brings the bait into close proximity to the prey tag. These two come together and they perform a function where they cut off this reporter molecule, and then it's released, and you can detect it as an indicator of A and B interacting. Okay? So one of the other common systems in two hybrid system is the one listed here, right? So this is called MYTH, which stands for membrane yeast 2 hybrid. So the cells you're using in this system are yeast cells, and the proteins you're studying are membrane proteins. So you're trying to look at two membrane proteins and see if they come together and interact. All right. Now, another method that also uses fusion proteins is called BioID. And this is an enzyme-based method that results in proximity biotinylation. It's very wordy, but here is what happens. So in this particular scenario, all right, you have one single protein of interest, and then you want to find all of the interactors for that protein of interest. So remember, with our two hybrid system, we were only looking at two specific proteins and seeing whether they interact. With BioID, you have one protein that is your protein of interest, and this method will allow you to identify all of the other proteins that will interact with your protein of interest, not just one single other protein, okay? So your fusion protein is going to be composed of your protein of interest, and you're going to add this enzyme called BCHA to the end or to the beginning of your protein of interest, okay? So what BCHA does is it will add a molecule called biotin to any proteins that come very close to your protein of interest. So within 10 nanometers of your protein of interest, BCHA sees that other protein and it's going to attach a biotin tag to that protein, okay? So then what you can do is purify those biotinylated proteins using affinity chromatography. So biotin will form a very specific interaction with another molecule called streptavidin. So you can take streptavidin, link it onto a column, run your cell lysate or your crude extract through the column. It will pull out all of the proteins that have been biotinylated, and those would be the interactors for your protein of interest, right? But here is a problem with BCHA, okay? BCHA, when it sees a protein that is very, very high in number, so it's highly abundant inside the cell, BCHA can actually add a biotin to those proteins even if the protein is not within the 10 nanometer distance. So this is just a quirky side of bird egg. So if you have very high amounts of a particular protein, they might get accidentally identified as an interactor, even though it's not actually interacting with your protein of interest. Okay. All right, the final method that uses a fusion protein is called resonance energy transfer. So, resonance energy transfer. And there are two versions of this. FRET, which uses fluorescence resonance energy, and BRET, which uses bioluminescence resonance energy. But the theory behind these two are essentially the same, okay? Both of these techniques are going to be used to determine whether or not two specific proteins interact. So similar to that two hybrid system, we are now looking at two very specific proteins, and whether or not these two proteins form an interaction, okay? So I'm going to start off with FRET, okay? So here are the fusion proteins you need for FRET. So what you do is you will link one of your proteins, let's say protein A, to a fluorophore, okay? And I'll call this fluorophore A, all right? And then you're going to link your other protein, in this case protein B, to a different fluorophore. I'll call this fluorophore B. Now, these are not just randomly picked fluorophores, all right? These are special fluorophores where the emission wavelength of fluorophore A is equals to the excitation wavelength of fluorophore B, which basically means if you have A right next to B, you excite A, right? A is going to emit light, and that light can be used to excite B, causing B to emit light, right? So that's the general idea of resonance energy transfer. You have transfer of energy in the form of light from one molecule to another, okay? Right, so if you have protein A forming an interaction with protein B, they come very close together, right? And they're going to bring the fluorophores that are attached to them very close together as well. So at this point, you're going to shine a ray of light onto your cells, and this light is going to be the excitation wavelength for fluorophore A, right? Fluorophore A gets excited, it's going to release light, right? So this is an emitted light showing in blue for fluorophore A, and this emitted light from fluorophore A is going to excite fluorophore B, causing fluorophore B to emit its emission wavelength, okay? So then what you're looking for then is the emitted light coming from fluorophore B. And once you detect that emitted light coming from fluorophore B, this tells you that yes, A and B are interacting. And once again, I apologize, this red one should be a B, not an A, right? So that is FRET. Now, BRET with bioluminescence is basically the same theory as FRET. However, with BRET, the fusion proteins look a little bit different, and you don't need an excitation wavelength. So you don't need to physically shine a ray of light onto your cells. So here are the fusion proteins in BRET, right? So you have, once again, a protein A and a protein B. We're looking at whether or not these two specific proteins interact. You're going to link to one of your proteins, say protein A, you're going to link an enzyme called luciferase, all right? So this luciferase is actually an enzyme that produces its own light, right? So it's constantly making its own light. So this is known as bioluminescence, right? Which is what the B stands for in BRET, because it doesn't need incoming light. It can make its own light, all right? And the light that is made by luciferase has a wavelength that is the same as the excitation wavelength for a fluorophore, which you attach to your other protein, right? So if proteins A and B interact, right, they come very close together. This brings luciferase into close proximity to your fluorophore, right? Luciferase is making light. This light is now able to hit fluorophore B. It's going to excite fluorophore B, and fluorophore B is going to emit its emission wavelength. And once again, this is what we are detecting in the experiment. Once you see the emission light coming from fluorophore B, you know that proteins A and B have formed an interaction, right? So this is Fret and Brett, okay? Now, I also want to just quickly talk about fluorescence microscopy in general. So this is the same microscope that is used for Fret and Brett. But with fluorescence microscopy, right, you can also use it to actually locate proteins. So determine the subcellular location of a protein. So in this case, your fusion protein would just be your protein of interest attached to some sort of reporter molecule, all right? So it could be a fluorophore, it can be luciferase, anything that can be visualized using some sort of method, right? So then you would see where that protein goes inside of the cell, and you'll be able to follow where that protein moves to because you will be able to see the reporter molecule, which is attached to your protein. Now, you can also use fluorescence microscopy for co-localization experiments. So you have protein A, right, attached to a reporter. You have protein B, which is attached to a different reporter, right? And then you're basically looking to see whether or not reporter A attached to protein A is going to overlap with reporter B attached to protein B, right? Now, co-localization assays are less sensitive than Fret and Brett. So co-localization assays is not a good indicator of interaction. So with co-localization assays, right, if you see reporter A close to reporter B, this tells you that your protein A is close to your protein B, right? But it doesn't actually tell you whether or not A is forming those non-covalent interactions with B. It just tells you that A is somewhere in the vicinity of B. So they're sort of close together and therefore their signals are overlapping. Whereas all of these detecting interaction methods are more sensitive, right? They require very, very close distance between A and B, and it's close enough that you're assuming A and B are probably forming non-covalent interactions if they're that close together, okay? All right, so those are protein-protein interactions. But obviously, proteins don't just interact with other proteins. Proteins also form interactions with other molecules that are not proteins. So for example, your ligands in your PDB assignment, all right? So one of the types of protein-non-protein interaction that is very prevalent in biology is the interaction between an enzyme and a substrate. Right? So enzymes will take substrate and then turn it into something else called a product. So when you're looking at enzyme substrate interactions, most of these are done in vitro, so meaning outside of the cells, simply because it is easier to monitor this interaction when you're looking outside of the cells, right? Because you get to control how much enzyme is there, how much substrate there is, and then we'll talk about the details of this when you learn about enzyme kinetics. But in essence, in order to look at the interaction between the enzyme and the substrate, what you're measuring is the reaction. So substrate being turned into product is an indicator that this enzyme is interacting with your substrate because it is able to then turn it into the product, right? So you can measure this reaction using two ways. You can either look at the appearance of the product, or you can look at the disappearance of the substrate. Which one you choose is going to depend on what the substrate is and what the product is, right? Some molecules are easier to monitor compared to others. So you can look at this appearance or this disappearance via stuff like absorbance, right, using fluorescence, or you can use a method called chromogenesis, where you're essentially forming a product that is physically colored. So for example, p-nitrophenol is a yellow colored compound. So once p-nitrophenol gets formed, the solution turns yellow, and you know that the enzyme is turning substrate into a product, right? Now, sometimes you're not so lucky. Sometimes the product is colorless and the substrate is also colorless and it doesn't absorb and it doesn't fluoresce. So in this scenario, in order to actually visualize the substrate or the product, so be able to detect it, you actually need to couple this to a secondary reaction. So it would take the substrate and then turn it into something else. And this something else can be, say, p-nitrophenol, and can be visualized using spectroscopy or just by your eyes, okay? So in summary, right, you can look at protein-protein interactions using pull-down assays and cross-linking, right? You can look at protein-protein interactions inside of cells using fusion proteins in two hybrid systems in BioID, as well as FRET and BRET, right? And you can look at the enzyme-substrate interactions by actually monitoring the appearance or disappearance of products and substrates respectively. So those are all of the techniques that are covered in the lecture regarding to protein function, right, which is not related to your PDB assignment at all, but you will need to know this for BBQs and your quizzes as well. All right, so I'm going to go back to the question sheet. All right, and we'll give it 1 second to load. All right, so once again, I have a table in this PDF file for the notes and cues, a table that summarizes all of the different methods for studying protein interactions. So if you prefer this particular layout, it's available for you, right? So now I'm actually only going to go through the first 3 questions because question number 4 and question number 5 are actually quite challenging. These are just questions that I made up to make sure you understand the concept of cross-linking, all right? And these cross-linkers, because that tends to be a concept that students get confused about the most. So I will leave you guys to try question 4 and 5 on your own, and then I'll post answers and explanations as to why that answer is correct for those 2 questions. So I'm just going to go through 1, 2, and 3 very quickly. These are very very straightforward, simple questions. Okay. So question A asks, which reagent will let you detect the presence of cysteine residues on the surface of a protein? Okay. So your choices are beta-mercaptoethanol, streptavidin, iodoacetamide, and SDS. Okay. So straight off the bat, SDS is gone. It has nothing to do with cysteines because what cysteines do are form disulfide bridges. SDS doesn't do anything to those disulfide bridges. Streptavidin, nope, that's used for purification when you have biotin labeled proteins, all right? Now, beta-mercaptoethanol is an interesting option because BME if you remember, was something that Dr. Patterson mentioned very briefly in a lecture from, I think, week 1 or week 2. And she mentioned this is a reducing agent, okay? And it's going to break apart disulfide bridges, right? But if you think about it, if you never had any cysteines or disulfide bridges to begin with, treating that protein with beta-mercaptan doesn't do anything because your result before treatment versus after treatment is completely identical because you don't have disulfide bridges or cysteines to begin with. Okay, so beta-mercaptan ethanol is also out. Now you are left with iodoacetamide. And remember, iodoacetamide is an example of a chemical crosslinker. And what it does is forcibly make disulfide bridges between cysteine molecules. All right, so I'm going to explain this a little bit more. All right, so if you add, okay, right, backtrack first. If you don't have iodoacetamide, right, you have protein, okay? Let's say you run it on an SDS gel, right? Remember, here is smaller proteins and here is bigger proteins. Let's say your protein is of size 50 kilodaltons, right? No iodoacetamide, no formation of disulfide bonds. So your protein is just going to be your protein by itself. Versus once you treat it with iodoacetamide, it's going to detect any of the cysteines that are present, force it to make a disulfide bridge with another cysteine molecule, which means your protein is likely now linked via a disulfide bridge to another protein or to other identical proteins. But nonetheless, that protein is now suddenly bigger in size. So I actually see a shift on your SDS page gel, right? Because now your protein is attached to something else via the disulfide bridge, and so it's of a bigger molecular weight. Now, what if you don't have cysteine residues, right? If you don't have cysteine residues, that protein isn't going to form any disulfide bridges. So you'll be able to tell because what you'll see is instead of this upward shift in size, you'll just see that your protein stays exactly where it is. It's still 50 kilodaltons because it's not being covalently attached to anything via a disulfide bridge. Okay, so that's how chemical crosslinkers work. All right, second question then. You've identified a novel protein, keyword here, novel, meaning new, all right? And you want to find other proteins that interact with your protein of interest. What technique do you use? So you want to identify other proteins that can form interactions with your own protein, okay? So here are your options: co-immunoprecipitation, bio-ID, Brett, and the two-hybrid system. Now, remember two-hybrid system? The question we're trying to answer is whether or not two specific proteins interact, right? So say protein A and B. That doesn't help you, okay? Because you have a new protein and you want to find other proteins that interact with your protein of interest. So you don't know what those other proteins are yet. You're trying to isolate them and then find out what they are. So two-hybrid system doesn't work because it requires you to know two specific proteins in order to analyze whether or not they form an interaction. Same thing with Brett, is also looking at interactions between two very specific proteins, all right? So Brett is out as well. We have co-immunoprecipitation and bio-ID. Both of these methods can be used to identify interactors, including unknown interactors for your novel protein, right? Because with co-immunoprecipitation, what you can do is, after you run the gel, you can cut out that interactor band and then throw it into mass spec to see, oh, this protein is CDE or whatever protein it is that's bound to your actual protein, right? Here is where the question is a little bit tricky, okay? So the key lies in this word here. It's a novel protein, so it's a brand new protein. Now, with co-immunoprecipitation, in order to pull down your protein of interest, you need an antibody versus your protein, right? An antibody that's going to bind to that protein. However, because your protein of interest is a brand new protein, chances are there are no antibodies that you can buy that you know for sure will bind to your protein. So this method means a lot of work. You have to actually make those antibodies yourself before you can do co-immunoprecipitation. So the easiest answer is actually bio-ID. All you need to know is the gene for that protein, right? Get the gene, add in BRA to the end, right? Get the fusion protein of the novel protein plus BRA, and then BRA will biotinylate anything that your protein interacts with. You can pull all of those proteins down and then throw them into mass spec to identify them, okay? Now, moving on to question 3, okay? The number of functional group targets in a protein is important when you're choosing a cross-linker. Which of the following would be the least abundant? This is actually more of a week 1 question than a week 3 question, but because it has to do with cross-linkers, it could potentially show up in your BBQs or quizzes. But your options are carboxyls, carbonyls, amines, and sulfohydryls. So these are also the thiol groups, so your SHs, right? So it's asking which of these following groups would be the least abundant, right? Carboxyls, not so much, right? There are a lot of charged amino acids that have carboxyls in their side chain. And you also have a carboxyl at the C terminus of your protein, okay? Carbonyls, even more abundant. You have a ton of carbonyls in your peptide bond, right? This guy here. Same thing with amines, right? You have amines in your peptide bond as well. You also have amines in amino acids like lysine side chains. You also have them at the N terminus of a protein. So still very abundant. Sulfohydryls, on the other hand, or thiol groups, they are only present in cysteines and methionines. So the answer would be D. This thiol group is the least abundant, okay? So once again, go over question 4 and 5 on your own. See if you can get the answer, and then I'll post the answer and the explanation a little bit later, right? So probably right after this session, right? So that's it for lecture material. And then now I want to talk about the PDB assignment, and I'm going to write this with you, okay? So this is going to be a sheet that I fill out during the session. And then once it's filled out, I will send it into group chat for people to look at. Okay, so you will get a copy of this. So all you have to do is just sit, listen, and follow along and see what I write. All right, you don't need to take any notes. All of this will be given to you. All right, so the reason why I wanted to actually cover this is because relating what you read in the paper and what you find out about your protein or your structure to content material that you learn in the lecture is something that Patterson adores. Right? She loves it when students make connections to what they've learned. And so by doing this, you're going to be able to get a really high mark for the assignment because Patterson will be very happy. And because she likes for students to make these connections, she will tell TAs to also look for these connections as well, all right? So it doesn't matter whether or not Patterson is marking your assignment, okay? All right, so I'm going to start off with an example in purification. let's say you're writing about purification of your particular structure, and then you see that it was purified using this column called source Q. So first tip, right? When you're writing part 2 and you encounter a very specific reagent or a very specific column that was used to purify, if you want to give this name, that's okay. But if you give a name like this, you need to also include what it is, all right? Because your TA might not actually know what the heck a source Q column is. So you need to Google this and then you need to write it into your assignment. So if you actually Google source Q, it's an anion exchanger. So in your write-up, you would have a sentence like, this protein was purified using a source Q column, which is an anion exchanger. So you're relating it to what you learned in ion exchange chromatography, okay? So this would be a sentence that you need to include if you are going to include the name of the column, the specific name of the column, all right? Now let's try to explain the pH a little bit. So let's pretend the authors isolated this protein, purified it using a source Q column, using a pH of 8, all right? Now, what do you know about ion exchange chromatography, right? So that would be the lecture content. So you know that an anion exchanger are positively charged and therefore bind to negatively charged proteins, right? For a protein to be negative, okay, the pH must be what? pH must be greater, greater than the PI value of that protein, all right? Now, if you actually know the PI value of your protein, wonderful, you can add that in there. But if you don't know the PI value of the protein, most cases, you probably won't be able to know the PI value of the protein. You can just say that the author have to use a pH that's greater than the PI value of the protein. And so therefore, the PI value of my protein must be what? Less than the pH of 8 that is being used, or else it's not going to bind to the column, right? So this is relating to what you learned in lecture. Now, if you're talking about ion exchange chromatography, if you have a space for it in your part 2, also talk about the salt concentration that is being used when that protein is first added to the column. Because remember that high salt is used to elute your protein, all right? So you want to indicate that this buffer contains low salt, right? 0.5 millimolars NaCl, low salt, okay? And the reason for this is because to ensure that the protein can adsorb, remember this word, right? You can put this word in here, to the column. Because high salt conditions will kick the protein off of the column. So if you have a salt condition that's, say, for example, 1 molar NaCl, there's a lot of ions around, your protein is going to be neutralized by those ions, and you're not going to be able to actually have that protein bind to your source Q column, right? So this is an example with ion exchange chromatography. Now, a lot of you will see that your authors used affinity chromatography instead. And I'm going to talk about the nickel NTA affinity chromatography column, right? So once again, if you see a very specific name, so in this case, an Ni HeLa chelating column, Google it, find out that it's actually a nickel NTA type of column. And obviously you would need to explain that in your write-up, right? So in this case, it was purified using a nickel HELA chelating column, which is a nickel NTA column for affinity chromatography. So this is how you make that connection to what you learned in lecture, okay? Now, let's say the authors used a pH of 8. You have to explain the role of the pH in the purification step. So why would the authors choose a pH of 8? The reason is actually completely unrelated to your protein, because remember, with a nickel NTA affinity column, you have a polyhistidine tag that's attached to your protein of interest, right? And it's this tag that binds to the nickel NTA, okay? And I have just included an illustration directly taken from her slides that shows you how histidine can bind to nickel. And you'll notice that histidine binds to nickel, right, using a nitrogen in its side chain, right? Now, histidine side chain is what? It's ionizable, right? So it can get deprotonated, it can also get protonated. If you look at the version of histidine that is required to bind to this nickel column, it's just nitrogen by itself, all right? So it actually needs to be deprotonated to bind to the nickel column, all right? Now, what is the pKa value of histidine? pKa value of histidine is 6 for its side chain, right? Okay, so why use a pH of 8? Okay, when pH of 8 is greater than the pKa value of your histidine side chain, the histidine side chain is going to be deprotonated, all right? And so having that deprotonated histidine side chain is required for it to bind to the affinity column. So if you use the pH of, say, 4, which is less than the pKa value of histidine, histidine side chain gets protonated, and you end up with a hydrogen on this nitrogen, and this becomes positively charged, and it is no longer able to bind to the nickel. So in this scenario, your protein would just flow through the column and you lose it. It's no longer bound to the column, right? So once again, you're including information about the pKa value of histidine side chains. You're including information about ionizable side chains, deprotonation, protonation. You're including info about the histidine tag, the affinity column. All of these are things you've learned from lecture. And you can explain it in very simple words using lecture material. So this is how you connect to lecture. So you don't need to explicitly say, as we learned in lecture, or this is related to lecture content. Don't say that. Just use what you learned in lecture to explain what you see in the paper or what you see in the structure, right? So these are examples for part 2. Let's talk about part 3, okay? Part 3, you have to talk about protein folding. Okay, so for example, how does it fold? I'm actually bringing it down here because there is a picture, right? So, give me 1 second to connect the charger. All right, so this was basically what I generated from PyMol during the summer for the students. So this is that electrostatic map you would have made using Icon 3D instead this semester, but it would look something very similar to this, right? So when you're talking about how a protein folds, right, the one thing you should remember from your lecture that's really, really important is the fact that protein folding is driven mostly by hydrophobic interactions. So hydrophobic interactions are the main driving force of protein folding. All right, so you will be able to relate this to your electrostatic map, because what does the protein want to have on the surface exposed to the polar water environment? It wants to have the hydrophilic amino acids being on its surface, and it wants to bury its hydrophobic amino acids inside of the protein instead. So you can say something like, protein folding is driven mostly by hydrophobic interactions, and as seen in, this would be your figure A, right? The surface of this protein is mostly composed of polar amino acids, right? You would explain hydrophobic interactions a little bit before this, saying that it wants to bury all the hydrophobic amino acids inside of the protein, and therefore it leaves mostly the hydrophilic, aka polar amino acids, on the surface of the protein, which is indicated by the colored regions. So if you see color, it's a polar amino acid. So you can see most of these are colored, but just a few hydrophobic areas on the surface, right? So this is another way you can relate to what you learn in lecture with this specific figure. Now, what about the spectrum figure? So once again, this was the one I made using PyMol from the summer, okay? So with the spectrum figure, you can visualize all the secondary structures. So all the alpha helices, all the beta turns, all the random coils, all the beta sheets, right? So if you see that your protein has a lot of beta sheets, for example, right? What you can do is talk about this being a secondary structure which are formed by hydrogen bonding between the backbone of different chains, or of different beta strands, all right? And what determines which secondary structure is being formed are the amino acid sequence, or what amino acids are in this particular region of this protein. Type of secondary, right? So if you see beta sheets, you could then hypothesize that beta sheets is an indicator that the amino acids in this region are likely bulky amino acids, which are your aromatic amino acids, and your beta branched amino acids. So these are your ICTV wife. Obviously, cysteine is out, all right? So for example, you can add a for example and give maybe two of them, all right? And T. So this is how you would relate the secondary structure to what you've learned, all right? You can talk about what you think are the amino acids that are involved in the folding and formation of these particular secondary structures, right? With alpha helices, you can talk about the different amino acids that are found in alpha helices. You can potentially talk about amphipathic alpha helices. If you see an alpha helix that's half buried inside your protein and then half showing on the surface of your protein, you can say that, oh, the surface that's showing likely contains polar amino acids, and then the other phase contains the nonpolar hydrophobic amino acids, right? So that's part 3, okay? Let's move on to part 4, and we're going to talk about the molecular interactions. These are the easiest to write in terms of connecting to course material, right? Because you are going to be talking about interactions that are formed between functional groups, right? Which is week 1, introduction week material. okay? So here is a zoom up of the interactions that are occurring in my protein with my ligand. My ligand is AMP or AMP-PNP, right? And I just throw a structure of AMP-PNP next to it, so I know which functional groups are forming interactions with which amino acids in this stick figure, okay? So if we just pick any random amino acid that's forming an interaction, remember we have to talk about all of these amino acids. So let's just say that, oh, I don't know, maybe lysine, well, lysine at position 372 forms a salt bridge with a phosphate on AMP-PNP, right? So when you're talking about the molecular interactions, you need to identify the amino acid, the position it's at, the type of interaction, okay, in this case a salt bridge, and what are the functional groups that are involved. So what are the functional groups? Lysine has what? A positively charged side chain with that positive charge on an amine. All right, so you have a positively charged amine group in lysine that can form this salt bridge with the negatively charged phosphate group in AMP-PNP ligand. All right, so this indicates to the TA that you know the properties of lysine, you know the properties of a salt bridge, right? So this is relating once again to what you learned in lecture. So you always want to talk about not just the amino acid forming what kind of interaction with your ligand, but you also want to talk about the functional groups that are involved in this interaction. So if instead you are talking about a hydrogen bond, you want to indicate which functional group is the hydrogen bond donor and which functional group is the hydrogen bond acceptor. And this is how you relate back to what you learned in lecture. Now, if you are lucky and you have a protein with a mutation in it, this part becomes even easier to write. Because let's say that, oh, I don't know, this same lysine 3 at position 372 got changed into a glycine molecule. And then what they see is that there is decreased affinity for ligand AMP-PNP, all right? So if you have that mutation, you want to explain the normal protein with this lysine, right, versus your mutated version with this glycine. And you want to compare the interactions they are able to form with your ligand in order to explain this decreased affinity that was observed or whatever the authors observed in your paper. Okay, so in this scenario, I would have already stated that lysine forms a salt bridge with the phosphate on AMP-PNP because it has a positively charged amine group that can form the salt bridge with a negatively charged phosphate, right? So now I also need to talk about my mutated version. So glycine is a non-polar amino acid with a hydrogen in its side chain. All right, what does this mean? This means that it cannot form interactions with the phosphate group, or just any group, honestly, with the ligand AMP-PNP, all right? So because it can't form interactions with ligand in this scenario, right, you lose that salt bridge, right? And then how do you relate it once again to lecture? Less bonds or less interactions means a weaker binding. In other words, a weaker affinity, right? So this is a reason why you would see a decrease in affinity for your mutant protein compared to the wild type protein, right? So make sure that you explain all of these little details as long as these details are related to lecture content, all right? So keeping things simple, include all the details that are related to lecture content. Don't overuse very scientific words. And if you do use them, you need to explain what they are, okay? And another point that I just want to point out because I just remembered, right? Acronyms. Right? A lot of you will have proteins that are being referred in the paper as their acronym name. So for example, the one I use, GYRB. Okay? If you're going to use an acronym, the first time it shows up in your assignment, you need to tell them the full name. So GYRB is gyrase B. And then for the rest of the paper, you can use GYRB, all right? In the summer, students have lost marks because they didn't include a full name for acronyms. So make sure if you use an acronym, you give them the full name in brackets the first time you use it, right? So that is all that I wanted to cover in terms of relating assignments to course lecture material. Right, so does anybody have any questions regarding either the lecture content, so determining structure and function, or about relating to course material? If not, then the session is officially completed. All right, so I don't see any questions so far. So I'm just going to make a few statements so it gets recorded. And I'll also make those announcements on the WeChat group as well. So as you know, when you have an assignment due, we always run an assignment session on a Saturday from 9 a.m. to 12 p.m. Toronto time, right? Now your next assignment isn't until a while, so it's still quite a ways off. So you don't have to worry about that now, which means we won't have an assignment session this Saturday, okay? We'll have that session when it gets closer to the due date of your second assignment. And depending on what that assignment is and how much you need to know from the lecture materials, I will either cover it 1 week ahead of the due date or 2 weeks ahead of the due date if you already covered all the stuff in lecture, all right? However, that being said, I'm happy to hold a live Zoom session this Saturday from, let's say, 8 a.m., all right, to 12 p.m., just as a Q&A session. So something like office hours, but specifically for the PDB assignments. So for students that have questions, or you have, for example, sections from your paper that you don't understand, you are free to just drop in on that session, bring those stuff with you. You can screen share. I can take a look. I can annotate on your screen, and then I can help to answer any questions you might have about the PDB assignment live. So I don't have to answer it via WeChat, right? So I'll ask in the group chat, see if there are a lot of students who want me to hold this session. If so, I will hold the session. I'll just sit here and then people can come in, ask questions. Once they get the answer, they can leave to write the assignments. And I will probably, if I'm running it, I will run it from 8 a.m. just so that the students that are in mainland China don't have to stay up too late for asking questions. They can start from 8 p.m. in mainland China time, okay? And then, well, we'll see how long that session needs to be. It might end early, it might run long, but I'll be there to answer any questions if it is something that you guys want. And also, in terms of sending me your assignments for me to look at and give comments on. Now, because there are a lot of students in this course, which is wonderful, okay? And because this assignment is so long, it's 4 pages, single spaced, I'm obviously not going to be able to read everything over the weekend before the due date, all right? So if you want to send me your assignment, it's going to be a first come, first serve basis. So whoever sends it to me first is the one that I'll read first. And obviously, if I run out of time, then the ones that I can't get through, unfortunately, I just won't be able to read and comment on. But I'll just briefly scan through it, give you a couple of comments, see if you're missing some bits of information, maybe you need to make more connections to lecture material, and then send it back to you so that you have time over the weekend to edit your work and then submit it on the Monday. So with that said, if you are planning to send me your assignment for PDB, okay, send it before the weekend. So your deadline is 11:59 p.m. on the Friday, Toronto time. And this is just to make sure that I personally have enough time to actually read the assignment and give you constructive feedback, and then get you to improve the assignment before you submit it, right? Now, if I don't get to your assignment, I'm really sorry, I apologize in advance. But what you can do is, for example, if you have just like a short few sentences that you're not sure about that you want me to look at, that's something I can do very quickly, right? I can look at your sentence and say, oh, maybe this meaning is actually a little bit wrong, or maybe you should add this to your sentence as well. Okay, so if I don't get to read your entire assignment, you are free to send me short excerpts. So keep it to like short little paragraphs that I can send comments or feedback on. And those you are free to do anywhere between now until, let's say, the end of Saturday. So 11:59 p.m. on Saturday, you're free to send me those little short excerpts because they're faster to read, right? So those are just some technical stuff out of the way, and we're officially done for this session. So thank you guys for coming to week 3. Remember to do your BBQs and your quizzes, as well as your PDB assignment, which is due on Monday. So don't miss the deadline, all right? There's a heavy penalty for late submissions. Explaining part 3 tables, rationale of construct. Why did they choose this structure, for example, bound to AMP, PMP? Okay, so I can actually use the example that I use, which was 3ZKB, I think, so that gyrates that I typed down there. Sometimes the rationale is very simple, but it ultimately depends on the paper, right? The authors of your paper. So for example, you can have something where there have been previous papers that have been already published that show the structure of my protein bound to ATP, all right? So my authors, the reasons why they chose to determine the structure bound to AMP-PMP is because they want to compare it to the published structure with ATP in it in order to understand how my protein actually cuts ATP, because that's what my protein does. It cuts off a phosphate from ATP and then uses it for something else, right? Yeah, so it's basically explaining why the 3D structure have those ligand bound to it. So what you'll see is when you're reading through the purification methods, a lot of the time, the authors will isolate the protein, right? And then they'll add in the ligand, the specific ligand they want, and then they co-crystallize the protein bound to that ligand, okay? So it's essentially asking you, why did the authors choose to add that specific ligand to interact with the protein, right? So my example was because they wanted to find its structure to compare it to the structure of the protein bound to ATP, right? Sometimes it can be as simple as just having no previous papers published on the structure bound to that specific ligand. So the authors are like, oh, hey, this is something that have not been published before. Let's publish it. Let's find the structure and publish something new. Okay, so usually the answer is actually quite simple, but it ultimately depends on the paper and the authors and what the authors are trying to do in that paper. So yeah, so I'll give you another example that I just thought of, and that relates to the inhibitors, right? So for example, if the authors in your paper are determining a structure in order to produce an inhibitor against that particular protein, right? They might need that inhibitor to bind to a specific area on that protein. So for example, if the protein is an enzyme, that inhibitor might have to bind to the ligand binding site on the enzyme, so the substrate binding site. So in my example, my protein is an enzyme that binds to ATP. All right. So if the authors determine the structure of the protein bound to ANP, right, which is a non-hydrolyzable version of ATP. So ANP or AMP-PNP is itself an inhibitor of ATPases. If the authors determine that structure, it can help them improve the design of ATPase inhibitors. So that could be another reason why that particular ligand is being used to find the structure for, right? Okay, so any other questions? Oh, the glutathione separose column. So I can tell you that glutathione separose columns tend to be affinity chromatography. All right, so actually, hold on. I'll find a picture for you. Give me 1 second. Okay. All right. Okay, so the glutathione system is related to a protein called GST, which is glutathione S transferase. And oftentimes what you'll see is for affinity purification, instead of adding a histidine tag, they'll actually add a GST tag to a protein. And then GST is an enzyme and its substrate is glutathione. So the enzyme binds to glutathione. So if you tag your protein with GST and then you use a column that has glutathione attached to the beads, when you run your protein sample, the GST tagged protein is going to bind to that glutathione and then get absorbed onto the column. So for most cases, we are talking about affinity chromatography, right? So I have never seen a glutathione ion exchange column before, but you probably want to double check. So how you can do this is look at how they elute the protein, okay? So if it's an affinity column, they will be eluting the protein by adding free glutathione onto the column, right? Free ligand to kick off the GST tagged protein, right? If it was ion exchange chromatography instead, they would have to use high salt or change the pH to elute. So you can sort of tell based on how they elute the protein exactly what type of column it is. But here, I'll just show you this, right? The picture of this affinity chromatography. I think this is a better picture, right? So GST binds to glutathione. You can use free glutathione to elute. Now, obviously, if it's for protein isolation, right, you would have GST being linked to your protein of interest, and then you can isolate all of that together. Okay, so double check your paper, see how they elute the protein, and then you'll be able to tell. Ah, I see. Okay, so after extensive washing with phosphate buffer saline, the protein was then cleaved from the beads with thrombin. Okay, this is really, really interesting because it's something that you guys haven't covered yet, or you will never cover actually in this course. But sometimes when you're using affinity chromatography and then you're using a fusion protein, right? You're adding a tag to the protein. What you can add is something over here, a specific amino acid sequence that encodes for a cleavage site. So you would, to elute your protein, instead of using the free ligand, you would actually add in a protease. And that protease is going to look at that amino acid sequence, cut it, and then your protein of interest gets eluted off of the column, right? Now, yeah, I see the reason why you think it's ion exchange because of the saline, which is salt, right? Saline is salt solution. However, if you look at the wording, right, it's extensive washing with phosphate buffer saline. So they're washing it with saline. They're not eluting it with saline, right? If it was an ion exchange column, if you wash it with salt solution, it's going to it's going to elute the proteins off of the column. But in this case, they're just washing. So they're getting rid of any of the unbound, not bound proteins off of the column. So in this scenario, that saline solution is a washing solution. It is not the actual elution buffer. And the way that the authors eluted the protein off of the column is actually using this protease called thrombin, okay? Don't quote me on that. I can't remember if thrombin is an actual enzyme or not, but I know that thrombin will cut at a particular site. So it will actually cut amino acid sequence or cause it to break apart. So this is the method the authors used to get the protein off the column is instead of free ligand, they just made a cut in the fusion protein and then took the POI out, okay? So glutathione, saccharose, definitely affinity chromatography, yeah. Okay, so this would be like the live Q&A session that I'm proposing for Saturday. It's just me sitting here, people coming in with questions. Maybe you want to screen share your assignment and then stuff like that. All right, so any other questions? All right, cool. So I think everyone's good for now. If you have any questions regarding either the lecture material or the PDB assignment, feel free to ask me via WeChat. I'll make an announcement about both potentially holding a live session on Saturday, as well as sending me the assignment to look over and give feedback on. I'll send an announcement out in the WeChat group. And I'll also post the answers to that 2 difficult questions at the end for studying protein function. Right, so that's the end of the session. Thank you everyone for coming, and maybe I'll see you Saturday. If not, I'll see you the week after on the Thursday for our week 4 session.